Professor Timpson, BSc, MBBS, PhD, FRCP, MFP, MRCP, DRCOG, and I love myself. Look at me, being so charismatic. This afternoon, I'm going to take you on an exciting adventure to explore how influenza can cause a new epidemic. Such as the H5N1 bird flu epidemic, or more accurately, influenza A virus subtype H5N1 epidemic. We are also going to explore the possible mechanisms for the transmission of bird flu to humans. So, let's travel to the rural province of Ayutthaya in Thailand, where H5N1 has been isolated.
Why are there so many dead birds on my field? It's going to be very difficult to harvest, and I'm sure people will not want dead chickens in their cereal.
Good afternoon.
A chicken? But why haven't you died like the rest of your species?
Oh, I'm also infected with H5N1. I'll be gone in a few days too.
H5N1? What's that?
It's what they call the latest strain of bird flu, but I don't know why it has that name. Ask a scientist.
Did someone say my name?
No.
The H in H5N1 stands for hemagglutinin. Which is a trimer located in the membrane of influenza viruses. It's an antigenic glycoprotein. This is what it looks like. There are 16 subtypes, and H1, H2, and H3 are all found in human influenza viruses. Birds, however, are hosts for all subtypes of hemagglutinin. Sorry about that.
But what does hemagglutinin do?
It binds to cyanic acid, which is present on the surface of target cells. This causes the virus to be engulfed by a target cell.
Then what happens?
Part of the target cell membrane pinches off and surrounds the virus to form an endosome.
But why doesn't the endosome fuse with a lysosome in order to digest the virus?
A very good question, especially for a chicken. The cell tries to do this. When the lysosome fuses with the endosome, it acidifies the interior environment in an attempt to destroy the virus. When the pH drops to 6.0, the mission will change, and the hydrophobic polypeptide, called HA2, buried inside the hemagglutinin trimer, is now exposed. HA2 is a fusogenic peptide. This means it can insert itself into the endosomal membrane like a hook and pull it so that the endosomal and viral membranes fuse, releasing the viral RNA into the cytoplasm.
Wow! But what about the N in H5N1?
The N stands for neuraminidase. Here's a picture of its crystallographic structure. Remember that the virus is engulfed by the host cell when its hemagglutinin binds to the cyanic acid on the host cell membrane. Well, neuraminidase hydrolyzes this bond to prevent the influenza virus remaining stuck to the cell surface. There are 9 subtypes of neuraminidase. Humans are hosts for N1, N2, and N8, but birds are hosts for all subtypes.
So H5N1 means hemagglutinin subtype 5 and neuraminidase subtype 1.
That's right. Very astute of you.
But how does H5N1 manage to outsmart my immune system? And why does H5N1 have the potential to infect humans?
H5N1 can infect humans? You didn't tell me that.
The tragic reality is that the influenza virus has the ability to undergo antigenic variation.
How?
There are 2 mechanisms, antigenic drift and antigenic shift.
Oh, how does antigenic drift work?
The hemagglutinin gene mutates slowly, giving rise to a different amino acid sequence of the protein. These mutations accumulate with time. H3 in a man now is very different to the H3 that appeared in the 1968 H5N1 pandemic. New variants that are not detected by the host antibodies become prevalent through natural selection.
So how does antigenic shift work?
This can happen when a human influenza virus and an avian influenza virus co-infect an organism that acts as a host for both human and avian strains of the influenza virus. For example, a pig. When this happens, there is potential for the genes of these viruses to mix and create a new influenza virus with new antigenic properties that enable it to infect a human. Human-to-human transmission of H5N1 has not yet occurred, but there is a serious risk that it happens. For example, hemagglutinin could adapt to have a better binding to sialic acid in host human cells. Hemagglutinin from avian viruses binds to sialic acid with alpha-2', 3' linkage. If it adapts by antigenic drift or shift, so it binds to sialic acid with alpha-2', 6' linkage, then it will bind to sialic acid in human cells. Human-to-human transmission may also arise from a substitution of lysine for glutamic acid at amino acid 627 of a type of avian virus polymerase called PB2. That's what my research involves.
Wow, it looks like you have a really boring job. Why didn't you become a farmer like me?
Or a chicken like me?
Because I want to know the truth about H5N1 and have an unnecessarily long list of degrees after my name. I have to go.
But what about the bird flu?
Don't be a chicken. Get over it.